What we will be doing today is rushing through, shouldn't use the word rushing through, but it will be quite a pacey presentation because we are, as ever, trying to cram a day school into an hour and a half. As some of you will have experienced before, I think we did the Myriapod and no doubt on some of the other events that you've been attending. But anyway, I will just go through, let's get this thing to move. There we go. I'll just start very quickly tell you who I am and then introduce today's proceedings. So I'm basically a freelance zoologist. I was working for the Wildlife Trust in Sheffield. I was curator at Museum Sheffield for 25 years and worked at the University of Sheffield over the last few years on various research projects. I have the honour of having been able to write for the British Myriapod and Isopod group a CD-ROM presentation about centipedes, millipedes and woodlice, and that's what we're going to look at today. Also, some of you may have encountered me in the context of harvestmen and dragonflies and one or two other things. But today I will be looking just at the centipedes. Brief introduction about myriapods in general. This is following the same format that we did when we looked at millipedes last October, so I'm sure several of you attended that. I will try not to duplicate too much of that, but obviously there'll be different people on this call, so I need to go through some of the basic introductory material about myriapods. But all of the information will be available afterwards as a YouTube video. So if I'm racing through, you can take your time, you can stop screens and see the information at your leisure, hopefully. So that should work. Also, as I mentioned in a moment, you can also buy this presentation from BMiG. So we'll be looking at general ecology techniques for studying these creatures. What is a centipede? A little bit about taxonomy and the general characteristics that we need to consider when identifying them, their life histories. Primarily, I want to focus on the identification characters of a number of British species. It will give you a good flavour. We're not going to cover everything. There are too many to look at, and some very tricky characters, very small things to be looking at. So we're looking at it in quite general terms, but hopefully act as quite a good introduction. I'm getting blinded by the reflection of the sun off my neighbour's house, so hopefully you can still see me if I pull the curtains. So there'll be a section at the end where you can ask questions as well, hopefully, so depending on how much time we've got left. So this is the CD that was available from the British Myriapod and Isopod group, and it's designed as a standalone talk. So you can buy it, you can put it onto your computer and work through it. All the information is on the screen, all the text, and it's primarily out there so that you've got access to my photographs as much as anything else. It's as a resource to put some pictures against perhaps a lot of publications which are just text and keys, just to help you see what the whole animal might look like and some of the details. So you can work through that at your own pace. I'm basically going to go and talk you through it today with a few additions. But any more information that you want will be on that CD, and so you can look at this later. The previous webinar was the exact same thing, but it was looking at the millipedes. So those of you who saw that, if you didn't see that, it's available there on YouTube, and I'm sure there's a link via the Tanyptera project to go to that on their YouTube channel. The CD-ROM for those of you who were last time, the CD is now no longer available from Nature Bureau because BMiG have purchased all of the copies, and so they're no longer available to buy from Nature Bureau, but you can still get them directly from BMiG at the or by contacting Paul Harding. The various web links will come up shortly. It's apologies to those of you who paid more for it, but it's now a bargain at £7, £5 if you're in person, plus a couple of pounds for postage. Obviously, in the current situation, we're not doing live events, so we can't sell you these things directly person to person, but we hope to do that come the summer and any events subsequently. So it's gone down in price. BMiG, just you know, I've got the T-shirt. BMiG. BMiG.org.uk for anything to do with myriapods and isopods in Britain, and there's now information about marine isopods being developed too. So anything you want to know, go to that site. You can find checklists of centipedes, their various resources. You can highlight individual species where you can find images and taxonomic information and links to NBN atlas and maps, etc. So do use that as a good resource. Various things about distribution and references for each species. Another great source for information is the Facebook group, I suppose, Myriapods of Britain and Ireland. People, that's the place to find quick news, anything that's happening. That's a good place where people will post information, ask questions. It's a good place for you to post pictures and ask questions, and that's a very instant way of contacting and getting feedback from experts and other enthusiasts out there. So I would recommend the Facebook group. If you need images and you just want some pictures of things, my Flickr page is also out there, which has got a lot of the pictures that we'll be seeing today if you want a closer examination of those. So finally, I'm mentioning now a little bit iRecord. iRecord wasn't a thing when we wrote the BMiG CD. We would recommend as a group that if you want to record centipedes, use iRecord. Steve Gregory, I think in particular, is a verifier, and he will be very happy to receive records on iRecord and check out any photographs that you've got on there as well. So do use iRecord for centipedes, very active scheme. So no, we're still a bit bright. Let's keep the curtains closed, shall we? So let's start by get certain things out of the way before we get any further. Everyone knows that centipedes have 100 legs, right? Centipede, it means it's got 100 legs. Well, not really. They never have 100 legs, not unless they've been injured, not unless they've lost some or had some glued on by some bizarre occurrence. They don't have 100 legs. It's almost certainly the thing they don't have. So if they've been injured, that raises the classic joke, what goes 99 bump? Well, that's a centipede with a wooden leg, isn't it? Well, no. Sorry, that's not true either, unless they've got 3 wooden legs. All centipedes have an odd number of pairs of legs. So the nearest they can have is 102. If they've got 51 pairs of legs, or maybe 98 if they've got 49 pairs of legs, so that they can't have really 100 legs. Lots of species have around 30 legs, some have 42 consistently, and then there are many others which have somewhere between 70 and 202 legs in Britain, but never 100. So that's it. It's the end of the seminar. You've learned everything you need to know. They don't have 100 legs. The key thing I think with that I want to get across really, I think, with centipedes, for me, it's not really a group of animals where I remember everything about how to identify everything in the field. It's a matter really of, and what I hope we can do today is recognizing there are key characters that you need to make a note of, and then by combining and comparing a group of characters, you can work out what you're looking for. You can obviously still use dichotomous keys and work through keys just as with any other group. But the nice thing with centipedes in some respects is you look at your specimen, you gather a bit of information on its antennae, its eyes, its legs, various things, and then you can compare and contrast what it is that you might have in front of you through a key like this in Tony Barber's excellent aid gap key. I can show you things on screen. I know I'm very small for many of you, but anyway, that's an excellent and recommended book for you. The similarity, there are a lot of centipedes look very, very similar. They're ostensibly identical in some respects, but when you get down and you get your handlings to them, and you look at these characters, it's a matter of comparing and contrasting against one or another. Some can be recognized in the field with the hand lens, but it is really recommended to take voucher specimens wherever possible for closer examination. Some of the characters you're looking for are very small. Some of them need perhaps clearing with chemicals under a microscope so that you can actually see features more clearly. And the bottom line with centipedes is there's a lot of counting involved. So it's how many of this, how many of that, and that is just generally done more easily in the lab under a microscope, making a note and comparing things. It's just not to say that everything needs to be killed. You can do this with a piece of cling film or a little melamine wallet or something of that nature to hold things still, look at the characters you're after, and then things can be released. But I would always encourage voucher specimens to be taken if you can, because of the changing nature of taxonomy and information as it develops. It's really very helpful to be able to look back at a specimen years later, but I appreciate not everyone wants to do that. So it doesn't preclude you looking at centipedes at all if you don't want to take dead specimens. Normally, if we were on a field course, I would show you some centipedes. I would have been out and I'd collected you a lot of live centipedes for you to look at under a microscope. We can't do that today, unfortunately. Obviously, it's great that we can all be together from all over the world, in fact, but I can't. I have got one here, but that's not very helpful to you, is it, really, that one in there, a tiny little clip. So I videoed you a couple this week. So we'll start the proceedings so you can actually see a live centipede. So hopefully this will work. Hopefully this will be clear enough on your screen. And these are centipedes. This is what they look like. So clearly, lots of legs, antennae at the front, twitching away, trying to find its way. This one's blind, so it needs its antennae, it's got no eyes, and legs at the rear end, which are very distinctive on these types. So these soil centipedes, very, very flexible, definitely a soil-based animal. You can see this one tying itself into a knot. So some of them more flexible than others, but again, there's this common feature here of them having lots and lots of legs. These have slightly fewer legs, but we're looking at things like you can see the antennal segments are very distinct there, so counting those is very helpful. The plates along the back, the structures at the back end and they're very fast things. They're predatory, so you see them shooting away. This is another species, and they're all very fastidious, very like to be clean. You see them cleaning themselves. If you ever want to film one of these things, give them time and they'll stop and wash themselves. But there we are. So that you can see the enormous jaws at the front, these forcipules, these poison claws, as they're known. Those were your centipedes. That's it. That's as close as I can come to showing you them, but give you a very a general sense of how these things live and look in the flesh. Deliberately kept that very very brief, but we can now look at some of the details which will help you hopefully as recorders to identify these things. If I can just increase the size of this for you. Right. Hopefully you have a full screen saying an introduction to the myriapods and centipedes. If not, someone will tell me. So this is the first page of the Myriapod group CD, and we move that down. Okay, so hopefully that's fine. As I say, some of you will have done, will have looked at I showed some of this for the millipede course. I don't want to bore you with that. I will go through the beginning part on myriapods and the introductory section fairly quickly. If it's too fast, you can look at the beginning part of the millipede course on YouTube, and where it's, I wouldn't say it was necessarily slower, but it's another option for you to look at to see some of the basic introductory stuff. So I'm going to rush through this fairly quickly. So we're trying to cram a whole weekend or at least day school into a couple of hours. So apologies if it's all a bit swift, but let's move us on. Right. So why would we look at these things? Looking at centipedes as we are today, but also the myriapods as a whole, very useful groups to start studying if you've been into just insects in general, or perhaps other ground invertebrates. Then they're a nice additional group to look at. There's a lot of good information out there that will enable you to work through these and become familiar and be able to record them. They're available all around the year. It's not something that's relying on sunshine. There aren't too many species. I think there are 60-odd British species of centipede that we recognise now, including some in heated greenhouses. The skills that you learn on these courses are very, very transferable to identifying all sorts of other invertebrate groups. So quite helpful from that point of view. They are well studied. So actually the data that we have is quite helpful, but also the data that you can add to that is equally adding to a useful body of knowledge and research. But there is a lot of information out there, and there are a number of people that are becoming more skilled in these kinds of groups who can help you with the identifications and other information. That said, there are also myriapods that are turning up new species all the time, certainly with the millipedes. There was a mad flurry of millipedes in South Wales recently, and likewise centipedes too are appearing new to Britain every few years. So there's a chance for you to certainly be finding new things to your region, new things to the country, and occasionally new things to science, which adds motivation and excitement really to a lot of what we do. There is an active recording scheme, so you have the support of that. Steve is very good at getting back to people and verifying centipede records. they're relevant in conservation terms. There are economic issues with centipedes which make them important as soil invertebrates, as a lot of soil invertebrates are. So don't need to justify them anymore, but they're an interesting group to look at. They're an arthropod. They have a hard exoskeleton with jointed limbs. It's the same with lots of the other arthropods. They tend to suffer from loss of moisture, so tend to be in moist conditions. But they have a lot of similarities to a lot of other invertebrates that are out there, but actually within the myriapods, the centipedes, the millipedes, the symphyla are all very, very different in their own right. So we will concentrate on the niceties of the centipedes today, just to show that in terms of the taxonomy. There's the arthropoda en masse. The top one there, that's all the insects. All the insects are in that top bracket. All of the crabs, lobsters, etc., and the crustacea there. You've got all the spiders and the arachnids and scorpions, harvestmen down here. The myriapods occupy all that central area there. They're very, very different. If somebody says, well, you know, describe a myriapod, well, there are so many different forms and functions and behaviors and life histories that it's not something you can just quickly describe. So quite varied. Within the myriapods, there are a number of different sort of subclasses, suborders, and various things, which divide them into their various different types. Again, we won't dwell on that much this morning. They were originally gathered together by Latreille in 1810 under this title of Myriapoda, and that actually also included crustaceans as well. So the woodlice got in there as well as crabs and all sorts, because they had lots of legs. Eventually, that was refined to being just the few groups that we see today: the Diplopoda, the millipedes, the Chilopoda, the centipedes, symphyla, and the pauropods. They are all entirely terrestrial that we're referring to today. All the centipedes, they will breathe by means of simple diffusion of gases through tracheae. There are various holes when air gets into the body and is dispersed around the body in very simple terms. They are all divided up into a series of very similar leg-bearing segments of one sort or another. And as I mentioned, you know, you think of 100 for a centipede, but I think centipedes, they start with, I think the smallest number is 8 full pairs of legs, if I'm correct. Millipedes will start out with 6 pairs of legs, but actually you'll get up to 750 pairs of legs in a millipede, more than your centipedes. So very, very varied, as you can tell. So they might all be called myriapods, but the number of those pods is very, very different and very varied. I won't go into the symphyla and pauropods today, I'm afraid. And moving through the millipedes, we're into centipedes. So the term, the order, it's a class. Chilopoda is a class for the centipedes, very distinct from the millipedes, the Diplopoda. Diplopoda was meaning that there were 2 pairs of legs per body segment. The centipedes, the Chilopoda, have 1 pair of legs per body segment, which is obviously very, very different. But a key element is that centipedes are carnivorous. The Diplopoda are detritivores, and they're feeding on decaying matter. The centipedes are actively carnivorous and predatory. How they go about that varies, but they all have these large poison claws at the front for biting and subduing their prey. So a very different lifestyle indeed. The name Chilopoda actually relates to this poison claw structure under the head, which is formed from the front leg pair. Now you've got these big fang-like structures, but the base of that structure forms a kind of lip, and the name Chilopoda means lip leg in actual fact. So you've got this lip part at the front with these teeth at the front and the front of the coxswain of that, and then these enormous biting, pointed, poisonous forcipules, as we'll see. They're called big fang-like structures for hunting and for their predatory lifestyles. I mentioned that they have only one pair of legs per segment. That enables them to be slightly faster. They're built more for speed rather than for power. The millipedes are very much a bulldozer with power. Their gearing, if you like, is for the torque is for pushing and pressing and pile driver methods getting through substrates. Centipedes are designed to be agile and fast, and the single pair of legs seems to be a means of providing that mode of locomotion. Also, when you look at the structure of a centipede, you can see these offensive weapons at the front. It's got often, not all have got eyes by any means, but they might have forward-facing eyes to help them to hunt, and very sensitive antennae for seeking out their prey. So it's offensive, predatory animal. The millipedes, when we looked at those, there are quite a lot of different subgroups within the millipedes, all quite different. There are slightly fewer morphological types to get your head around with the centipedes. In Britain, really, we're only looking at 4 main types. The Scutigera morpha, and I will be using scientific names. There aren't many common names for centipedes. There is a list of common names should you wish to use them, but I will be referring to things with scientific names today. I will be repeating a lot of these group names, these order names later, so don't worry if you can't get your head around them initially. There may even be a quiz later. But initially, looking at the Scutigera morpha, they're very, very distinctive. They're like really big, very leggy spiders, very fast, the house centipedes. And in Britain, it's not really a thing that we need to concern ourselves with much. They occur. Scutigera coleoptrata is fairly frequent in the Channel Islands, but not desperately common. And there's also been this Theruonema tuberculata has also been found in the British Isles, but very, very distinctive and very unusual. And usually the records are associated with imported materials of one sort or another, but they do crop up from time to time. But we'll look at more of these shortly as we go through. The next main group are the Scolopendromorphs, the Scolopendromorpha. These all have 21 pairs of legs, and these include the large, poisonous, tropical, mean-looking, really. They get up to about 30 centimeters long in some cases, very substantial creatures, probably some of the largest invertebrates in the world, really. They have 21 pairs of legs and quite a bite on them. We'll be discussing that shortly. In Britain, that's only represented as a group by Cryptops, as a genus, the genus Cryptops. All of ours are Cryptops, so the names are fairly straightforward to remember. So all our Scolopendromorphs are cryptops. And then there are the lithobiomorphs, which look very, very similar. Lithobiomorpha, largely we sometimes call them stone centipedes, usually these chestnut-coloured, brown, but very, very similar looking species, which are quite surface active and probably the most familiar to most people, and they just have 15 pairs of legs. And finally, the Geophilomorpha, the earth centipedes, sometimes called wire centipedes, occasionally called wire worms. And these are the ones which have the majority of legs. They have somewhere between 35 pairs and 101 pairs in Britain. And so never less than 35 pairs. So a very, very leggy creature, and also a very, very flexible creature. These are much more a creature that's deep in the soil rather than running on the surface. Their hind legs as well, if you saw in the video I was showing earlier, the hind legs look almost like antennae and function very much that way. You can't always tell front from back. And that's probably the case with quite a lot of centipedes. There's this very much front end, back end is very, very similar, but particularly so in the geophilomorphs, but more later when we look at them in a little bit more detail. So because you've got these different forms, you've got these different levels of flexibility. The lithobiomorphs, these stone centipedes, are designed to be non-flexing. If you want to run fast on a surface, the last thing you want is to crumble from side to side. So there are various physical structures which help that those species remain in a sort of straight line, which gives them the ability to hunt at speed and be surface active. They're also they're a larger body mass in some respects than other centipedes, other than the Scolopendras, and that helps them because of the ratio between the size of the body and the surface area. They lose water less easily, and that allows them to be more surface active. The Scolopendromorpha, the cryptops species, fall somewhere between the real flexibility of the geophilomorphs and the rigidity of the lithobiomorphs. So they're quite active on the surface. They're able to run very quickly on surfaces, but they've got that little added element of flexibility that the geophilomorphs have. So we'll look more in detail at these as we break down and look at the groups for how to identify them in a few moments, but we need to press on. In terms of ecology and collection, again, I'll go through this rapidly, but it's hard to generalize, but they mostly live in damp places. As with the millipedes, they don't want to lose moisture, so they like damp places. The centipedes additionally are feeding on things which also like moist environments. So as a hunter, you'll find the hunter where his prey is. So looking under stones, looking under logs, among leaf litter and into the soil, that's where we'll find centipedes. Turning over soil, digging. Some of them are very, very subterranean, so digging down into soil does produce results in terms of finding some of the soil, more soil-focused geophilomorph centipedes. They are looking for a range of food types. Usually something that's alive, smaller than them, is usually the way that it's easiest to describe what a prey item is for these, but particularly woodlice and springtails form quite a large part of a centipede diet. So the younger manca, which is the name for a very young woodlouse, also more soft-bodied, easier to feed on, but mites, springtails, juvenile, centipedes, so they will feed on other centipedes. So if you're collecting centipedes, don't drop all your centipedes into the same tube, because they will, you won't necessarily just end up with one big fat one, but that's the gist of it. They are predators, and as with carabid beetles, you probably wouldn't throw a lot of predatory carabid beetles into the same live pot if you wanted to examine them later, because they will eat each other. So same with centipedes, they will devour each other if the opportunity arises. Largely subterranean, but let's say some things will be trotting across surfaces. The house centipede very much running around the walls of your house and hiding under window ledges and the like. If you should see those, certainly in the continent and continental Europe, they're much more common, and you see them much more actively. If you don't like spiders, you won't like Scutigiromorph house centipedes. It's the spider of nightmares. Reproduction with a lot of these, with all these species, keeping laying eggs. They're laying eggs, and whether they protect them and whether they just lay the eggs varies between the different groups, and we'll look at that as we go through. At the end, I've got a summary of the difference between some of these things. But in general, if you want to find them in these damp places, look underneath, pick them up, pick up the substrate, break off whatever it is you're looking, look under the bark, knock them into a tray, sieve, whatever it is that, if you've got a pair of forceps, pick them up. The key thing, I'll just check, the key thing with centipedes is they're very fast, and as they run away, probably the easiest thing to do is a wetted finger on top of them as quickly as you can, and stop it as it runs away, and then pick it up. More often than not, you'll do that, and certainly with some of the faster Lithobius and Scolopendras, you may just end up with their back legs. They will jettison their back legs. Keep them, because you may be able to identify the species just from the back legs. You may have lost the whole animal, but the back legs may just hold enough information. If you're very keen to know what it was, those back legs may just tell you sufficient to be able to identify the centipede. Otherwise, pair of forceps is potentially the easiest thing. Certainly if things are digging themselves down into rotting wood or into the soil, a pair of either flimsy storksbill forceps or a more robust fine pair of forceps, and tease things out of the gaps that they're getting into. So that's how you're going to catch them. The easiest thing is to probably just knock things into a tray so that you've got them contained. Sieving works very well. Centipedes, being the soil dwelling geophilid centipedes, they really don't like the daylight. They don't like heat. They don't like light. So if you put them in something like a Burlese funnel or a Torgren funnel with a light above, they'll avoid the light, they'll avoid the heat, and they'll work their way quite rapidly through, probably more so than a millipede would, through a funnel and fall out at the bottom end into your preservative fluid. And so the centipedes that are very surface active are very quickly spotted because they'll move quite quickly, but the Geophilus types will work their way through quite quickly. Actually, if you've got a sample of soil, it's remarkable how quickly they get down into it and disappear. Sieving works a treat if you've got a white tray or even a black tray, or as I've got a black and a white tray, so you can see pale things and dark things differentially in the tray. But sieving's great if you can afford a variety of different gauges of sieve. You'll get the soil centipedes falling through quite quickly, out of the bottom, and you'll get the larger centipedes, the stone centipedes, running around on the surface of the finer sieves. So that works very well. Pitfall trapping works well for centipedes too, but only really for those more surface active. The ones that dwell in the soil aren't particularly going to be running across and falling into your pitfall trap. But pitfall trapping does work quite well for, certainly for the Lithobius and the Cryptops species. In terms of preservation, when we collect, if you want to collect the specimen and look at it later, we would normally just drop it into 70%, 70 to 80% alcohol, industrial denatured alcohol, IDA or IMS, depending on what you want to call it. If you want to look at the DNA later on, ethanol, a higher percentage, 90–95% ethanol is preferred in terms of preservation of the DNA. The other thing, if you're looking, if it comes to that and you're interested in DNA, also chill down the specimen in a freezer as much as you can as well. Unlike perhaps an insect where you collect, you kill, you relax, you pin, you remove the pins. Yeah. With myriapods, the whole process is about dropping it into alcohol and it will stay there, and that's it. It's then the process is complete in some respects. You just need to label it. So preservation and long-term the conservation of the specimen is undertaken at the point that you kill the specimen. And I appreciate that not everyone wants to kill these specimens, but if you want to vouch a collection, just drop them into alcohol. We would recommend labeling includes the location name, the date, your name as collector, if possible a grid reference. Any other information such as habitat should go on the label as well as a minimum. I know a lot of you will have heard that stuff before, so I will not labour that point too much. So Myriapod group, a lot of resources out there. The bulletin comes out just about annually. There's also a newsletter that comes out twice a year. Good way of keeping informed, as well as on the Facebook group, which gives you more up-to-date news in relation to things. You won't be able to perhaps see this. I'm probably able to say probably a tiny little thing on your screen, especially if you're looking on a phone or an iPad. But classic work is Ted Eason's book on centipedes, the old Wayside and Woodland book. Absolute mine of information, and the classic. But Tony Barber has also, in more recent years, produced a Linnaean Society synopsis, which is excellent with a lot of detail. I think for today, again, I'll mention the aid gap key from Field Studies Council has got as much information as I suspect the majority of you would like. So that is the recommended text, I think, for this. In terms of biological recording, again, this slightly out of date on the CD, but do use iRecord. So centipedes specifically, again, looking at their life history, reproduction is only partially understood. There's not a lot of knowledge. I mean, certainly you look through Eason and he quite openly admits that some of the information is a bit vague. But most of the reproductive organs are situated at the hind end. The males produce a capsule of sperm, which they may leave behind on a pad of silk. The female retrieves that, picks it up with some modified limbs, and she will then take that away and may fertilize herself with that at that point, or may overwinter and use that sperm to fertilize her eggs the following season. So a lot of this, the reproduction is taking place in perhaps spring, summer, autumn, and over winter she may retain that package of sperm and produce her eggs later in the year. Again, I won't go into too much detail on this, but Geophilomorphs and Scolopendromorphs, they lay their eggs in a little cavity of one sort or another, and they will look after them. They're a very good parent and will protect. And until the individual offspring are able to sort of fend for themselves and go off into the wild. If you find that happening and you find a coiled up centipede around its eggs, I would recommend leaving it alone immediately, unless you're really keen to record. Quite often you disturb, I've certainly found a cryptops curled around its eggs under a piece of wood in a little chamber, and within seconds she'll start eating the eggs. So they will very quickly and very easily cannibalize their own eggs. So the less disturbance of a protected mother around her eggs, the better. I'm just brushing that. Remember on the front cover of the French guide, Les Myriapod, there's a very nice picture of a Scolopendra, which you may or may not be able to see, protecting its egg clutch. But if you disturb that, then there's every chance that they may eat them. So, but they'll protect that for a while. They'll look after their offspring and release them. The lithobiomorphs will just drop their eggs randomly around, and they fend for themselves, so that there isn't that same level of parental care. They will protect them a little bit by washing their eggs, and they can impart some protection from bacterial infection. And also with the lithobiomorphs, they'll coat them with sort of sticky substance which allows soil to adhere. So they become disguised. The eggs look like another particle of soil before they leave them. So they're not just randomly flinging them out there. They are at least giving them some kind of camouflage. They usually hatch and then produce a few larval stages, depending on which particular type of centipede we're talking about, over a few weeks. And as they mature, centipedes, depending on which group they're in, they mature slightly differently. And with the lithobiomorphs, they will have fewer antennal segments. They'll have fewer eyes, ocelli in their eyes. So those are not very good for identifying the species because they're immatures. Whereas something like the geophilomorphs, geophilomorphs are hatched with the right number of legs from day one, which is very helpful for identifying them, even when they're very small. So we'll summarize that at the end. So in the lithobiomorphs, they'll be adding legs, and they'll get 15 pairs of legs eventually, and they'll be adult, and they'll continue to pass through some stages to maturity, and they will continue to add additional characters until they are fully adult, at which point you can identify them. So beware. You can find a specimen and you will be thinking, you know, I think I know what it is, but actually you need to be very cautious as to whether you've got lithobiomorphs, gephyllomorphs, which different characters develop over time and which ones don't. As I say, we will summarize that at the end to make that more straightforward for you to remember. Just the pictures at the bottom there indicate how with lithobiomorphs, they're often this sort of chestnut brown color. They're very similar, reddish brown color. But when they've freshly molted from one stage to the next, they're often quite a pinky purple, quite an attractive color, but also a little bit softer, a little bit more vulnerable probably to predation. But if you find a purple centipede, it may well be that it's just freshly molted. Going on to how they feed, just quickly looking at these poison claws, these forcipules at the front, which is obviously a very key element of how they catch their prey. They do produce a venom, but also you bear in mind, if you're quite a small creature and these things are physically going to kill you anyway, by their sheer size of them is penetrating the body, is probably quite a deadly thing in itself, but there's also a poison involved. Certainly some of the larger centipedes will kill things within minutes. It's interesting, I once watched a very large pet Scolopendra that someone I had knew. We threw some crickets into the tank. It was at a public event at the museum, and I would say within 2 seconds it had caught 3 crickets. I mean, like lightning. It had just got it raced around, grabbed one, bit it, passed it down its legs, bit another one, passed it down its legs, bit another one. The third one it still held in its mouth. One it held somewhere in its mid-body, and at the tail end, the back legs were holding on to the third one. And I genuinely think that maybe take 2 seconds, 3 at the most. 1, 2, 3. It had just gone boom, 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 and caught 3 locusts, these crickets, and it was the most terrifying thing I've ever seen. But it was fascinating to see it grab and pass. So it was simultaneously passing its prey item to the rear end of the body while still chasing with these other limbs. So if you've ever wondered what's the value of having a lot of legs, it's because you can still walk, you can still handle things, you can still be biting things, you can still be hanging on to things simultaneously. I wish I had a video of it. It was quite a fascinating thing to see. But then it just sat there quite happily for the rest of the afternoon, munching on the one at the front, and then when that was finished, it brought another one up and ate its way through its 3 snacks. So centipedes, incredible predators, and yeah, I've not seen what the soil centipedes feed like, but certainly these Scolopendras are quite a thing to behold when they're feeding. Generally speaking, unless it's the British ones aren't really big enough to puncture human skin. That said, there is a species called Cryptops anomalans, which is perhaps 50 millimeters long. I once saw one in Sheffield. I saw 2 in Sheffield when I turned a stone on a guided walk. I knew it was new to the north of England, actually. I'd never been seen up here before. I basically said to the people on the guided walk, oh, these are new to Sheffield. I really need to catch these and it will bite me. And they sort of, what? So I thought, never mind. I went under, turned the stone again, grabbed these things. They immediately whipped round and bit me on the side of the hand, and I can tell you exactly where it bit me. That hand just there, because there were 2 little marks on the side of my finger and a little swelling for the rest of the day. But didn't really hurt, didn't do me any harm. And there are very, very few confirmed really harmful encounters with these species around the world. I think there's certainly a confirmed death of a child from being bitten by a scolopendra, but in Britain, you're fine. I'm going to move on to identification. This is what we're all about. Sure, this is what you want to hear. So I believe this seminar was scheduled to 2:30, so we've got half an hour left, so we should be all right. Feel free if you need to disappear. It was lovely to see you. So we're looking through for identification purposes, looking at the key characteristics. Scotigeromorph, very easy to identify. As I say, it's quite a big beast, long legs, 15 pairs of legs, and only 7 like shield-like plates along the back. So you can see these large purpley coloured striped plates along the back. They don't correspond directly to each leg. So there are 15 pairs of legs, but 7 of these tergites along the back of the body, and then they're quite cylindrical, quite curved. The legs are enormously long, and they're twice as long at the back as they are at the front, and they're fascinating things. And you try and catch one of these, and if you want to get a specimen of this with all its legs, good luck. You go near one of these, catch it, and they jettison their legs so easily. But they will regrow, which I guess is why they're not unduly concerned that they lose their limbs. But obviously, as a defense mechanism, if you grab a leg of one of these things, it can escape and they will regrow. I don't know quite how many it will regrow. I should imagine there's a limit. But yeah, they're fascinating things. They've got compound eyes. They cellular in such large groups that they effectively look like the compound eye of an insect. But very, very distinctive, very fast things. As I say, really only on the Channel Islands in the British Isles. So yes, we've got the lithobiomorphs, the stone centipedes, usually very similar in appearance. This time, unlike the scotigeromorphs, the number of plates along the back matches the number of legs. So if you're not sure, when we're talking about which tergite, which plate along the back we're talking about, or you can't quite work out, because they're sort of fat and thin, and they're fat and thin, so they vary in size, and you can't always, if one overlaps another, you can't quite always count them that easily. Count the legs, because the leg will correspond directly to the segment. So if you're looking for the seventh tergite, count the 7th pair of legs, and then you'll be able to see it there. So 15 plates along the back and 15 pairs of legs. They do have a variable number of antennal segments. So depending on which species you're looking at, count each of these different articles in the antennae. That's a really useful character. Somewhere between 19 and 54 can be quite varied, but a very useful thing to count. So it's one of those things, first thing you're doing is counting antennal segments. It may be that it's a really consistent number. Lithobius crassipes, Lithobius curtipes have 20. Lamictes has 25 consistently. There's not a lot of variation. So count the antennal segments. Young Lithobius do add segments as they grow, though. So be cautious that it may not be an adult in all cases. Also, some of the other characteristics are also being added, as I mentioned. So we'll look at those in a little more detail in a second. So you can get misleading figures because it's not quite adult. I guess the thing to do is collect as many from a site compare them and try and, you know, collect the adults where you can. But the more you've got, the more likelihood you've perhaps got of getting an adult. But also, if you're unsuccessful, go back later in the year, and you may be able to identify the thing more accurately later on as it matures. So each of the eyes of centipede, I may interchangeably call them ocelli or eyes. I know they have two eyes, one on each side, but the number of ocelli within that is what I'm referring to here. So lithobiomorphs have eyes, the geophilomorphs don't. Neither do the cryptops species. So eyes, we're really only interested in eyes with lithobiomorphs. They do vary from one eye on each side, one ocellus, to perhaps 40. Some of the large centipedes, sort of Lithobius pilicornis and forficatus, got a lot of eyes, and you are counting how many of these ocelli form that eye, that structure, and they vary in size as well. So the form of that the eyes take is indicative of a species. You might have one big ocellus at the back with 3 rows of smaller ones in front. You might have one big one and a medium-sized one and a few more little ones in front. There are some species like Lithobius curtipes where you might have one big-ish ocellus in the back of the eye, like here. So one larger one at the back of the mass of ocelli, and then the eyes, the ocelli in front might be in a circle with one in the middle, like a rosette, and that's what Lithobius curtipes looks like. Whereas Lithobius, other species, they might be in rows, they might be in all sorts of patterns, but anyway. So as it matures, it adds eyes up to a sort of finite point of adulthood, and there can still be a little bit of a range between one and the other. I mentioned, again, you might not see it on the small image here, but if you're using something like Tony Barber's Centipede 8-gap key, and you'll have a chart. So you've counted the antennal segments, you've counted how many ocelli there are in the eye. Just make a note of these things, and then you can work your way down and find within a range of this number of eyes, within a range of this number of ocelli, of antennal segments. It narrows it down to these up here, and then eventually the more characters that you identify and you look at and count, you narrow down the species. It's a very nice way of identifying a species, really. Just gather together the information. The other nice thing about doing that is what you'll find with centipede keys is that you'll work your way through a dichotomous key, and they'll ask you for something which requires you to look at the back of the animal. The way to examine under a microscope centipedes, a very good way, you could use a bit of cling film or something if it's alive, but in a preserved specimen, between 2 glass microscope slides, with just a slight amount of pressure, just to sort of flatten out a little bit, enables you to look at the top, then flip them over and look underneath, and then turn them back and look again on the top if necessary at the different characters. And I say that because you go through a key and it'll ask you how many, or what, to have a look at the tergites on the back, and then the next thing will ask you to look at some paws underneath. And then the next character, it'll ask you to look at something else, which will be on the top, and then something else underneath. And you do tend to be flipping centipedes quite a lot under a microscope, I find. So it's quite nice to look at everything that you can see from above. How many eyes has it got? How many antennal segments? Flip it over, look at its forcipular teeth, look at the paw punctures, whatever. Make a note of them, and then you can look at the key, the chart, and work out what that combination tell you as to what the species is before you constantly flipping back and forth. That's my tip for the day, if nothing else. So you counted antennal segments, you're looking at the eyes, the ocelli. And I mentioned in these forcipular coxosternites, I've described these to you so that you know what you're looking for when you read this in a key. Where these poison claws are underneath the face, underneath the front and the mouth parts of the centipede's head. The first modified segment with these poison fang-like claws on the front is called a forcipule. The base of the leg is called the coxa, and so the sternite, the sternum of the animal, the coxosternite, is the coxa on the front of the animal at the base of its leg on the forcipule. So a forcipular coxosternite is this bit here. So you've got, this is your forcipule, the biting fang-like part. This is the coxa of that structure. And so it's formed a sort of flattened sternite, and the leading edge of that has some teeth on it, has some bumps of one sort or another. And they can be counted, and that's a very useful character to help you identify the thing you're looking at. It may have, more often than not, there'll be two on each side. So you've got, that would be described in the keys as 2 + 2. Quite often you'll see these things defined as 2 + 2. So you've got 2 on this side here. If you can see where my cursor is on the image, you've got 2 there, and then you've got 2 here. So that's 2 + 2. On the one above, you've perhaps got 1, 2, 3, 4, 5, 6, 1, 2, 3, 4, 5, maybe 6. So that might be written as 6 + 5 or 6 + 6. And it's just these little teeth on the front edge. So basically where these sort of fang-like structures come, just in between there are some teeth. Really helpful characteristic that you're looking for. You may have up to 7 on each side, but that's quite characteristic. You may also be looking at the way that they sit at the front edge. So again, looking at this one here, the edge of this coxosternite falls away quite rapidly. So you've got the 2 teeth, and then it just drops away quite quickly at the edges either side. Some of them have a much broader shoulder. So you've got 2 teeth and then perhaps a broad shoulder. It might still continue out sideways rather than just dropping away. These are useful things to observe. They're very small, and it sounds a bit fiddly, but actually you've got your centipede, you're looking, you've counted its antennae, you look at its ocelli, you flip it over, you just count these teeth. It's relatively straightforward, but you can see why you need the specimens in your hand, really. I mentioned knowing which tergite you're looking at on the back. Let's say, in case those of you not familiar with the terms, this is your sternum and the front. You tend to think of sternum. So those are sternites, and the tergites are the ones on the back of the animal. So if you can't quite remember which one it is, it's not on the sternum, it's the other one. So it's on its back. And they alternate as they go along between a sort of thin tergite and a fat, bigger tergite, which is, several times fatter in that case. So large, thin, large, thin, large. And the hind corner of that may be rounded. So if you look at where my cursor is going, that's a nice rounded edge. These are nicely rounded. All of these have got nice rounded back corners. So there's no projection. Looking over here, this one's got a clear point at the back. This one here has got a clear point. So this is perhaps 9, and this is number 11, and perhaps 13's down there. And 7, 9, 11, and 13 may or may not have this projection. That's a very useful thing to be able to recognize. Sometimes it may be more of a squared off corner, which may be, you're not sure, but perhaps the combination of other characters that you looked at might help you to identify what you're looking for, and it might not be so important to know quite whether the corner was angular or not. But a decent microscope and adjusting the light can usually help. But these are all rounded, and this one has got a distinct point on it. So that's a very useful character too. So there's your classic. You know, if they did that every time, you'd be laughing. That'd be great. If you could just count the antennal segments, it'd be very easy. If you could see the eyes very easily. But those are the chief characters, which will narrow it down for Lithobius down to just to 2 or 3, and very often will tell you which species it is. So it's very straightforward. There are other characters which are slightly subtler, but which will help you with some of the more difficult things, which will help you. It's a bit more specialist, but this is the legs. So for those who are not familiar with the terminology for invertebrate limbs, first segment is the coxa, the one right next to the body. And in the case of a centipede, it's quite large along the side of that hind. This is the last 15th segment in this case, quite a large coxa. You've then got a small trochanter. You've then got a prefemur before the femur. So it's not just a femur, you've got a prefemur, which is quite large. You've then got the tibia. You've then got a tarsus and a metatarsus. So rather than just a single tarsus, which might be in case of some groups, you've got a metatarsus. And in the case of something like Scutigera, that might be broken down into lots and lots of different segments and lots of metatarsal segments, but generally it's this one. And then right at the apex, there may or may not be a claw. In Lithobius, there'll be a claw of one sort or another, and it may be just a single claw. It may have a little accessory claw. It may have 2 little accessory claws. So again, looking at that, looking at the end of the claw, seeing whether there's just a little extra spike, a smaller claw, that's quite helpful, but does require a decent microscope. Can occasionally see it with a hand lens if you've got a decent hand lens. So that's looking at Lithobius there, but also with the great thing, this is where it gets a little bit more specialist. I won't labour the point too much, but all Lithobius species, and that's not all the lithobiomorphs. Lamictes is the only lithobiomorph that we've got, which is not in the genus Lithobius. So it's easy, they're all Lithobius apart from a couple of Lamictes species. Lamictes doesn't have any spines on the leg joints, so that's an easy and really helpful character to know that you're looking at Lamictes. It also only has 1 ocellus on each side, so Lamictes is a fairly straightforward creature. But all the other Lithobius species have different spines around the ends of the joints, and these are all mapped out. And if you're, say, fortunate enough to have a copy of Eason, quite ridiculously, really, every spine is mapped for each of the Lithobius species. So the actual pattern of spines, spinulation, if you like, is actually recorded on there. Now that's too much detail for most of us, but it exists if you should need it. But there are 1 or 2 key characteristic spines which will help you to identify certain particular species of centipede. And the chief one probably, and the one I'll describe now, just so you understand the terminology, is this 15 vac. And the 15 vac you may encounter in a key. It will be describing it, and again, in Tony's key, there is a column here as to whether the 15 vac occurs in this species or not. So 4 species or 3.5 species may have that extra spine. But I'll explain what that means in case it's ever confused you, and then you can work it out what any of these characters mean. because you know what the pre-femur, if I just go back one to where we were, you've got CTP, FT, you've got the coxa, you've got the trochanter, you've got the pre-femur, the femur, the tarsus. Those are the things which those are the key articulation points that you need to be looking at. 15 refers to it's the 15th pair of legs, and invariably it's 15th, occasionally might be the 14th. But generally it's the characteristic spine that you're looking for is the 15th. V means that it's ventral. Dorsal, as in dorsal fin of a shark, you know, dorsal is at the top of the back. So ventral is beneath. So it means on the beneath the 15th leg. A means anterior rather than posterior, so it may say A or P. And then C is referring to the fact that it's on the coxa. So on the 15th leg, ventrally, on that leg, anteriorly at the front, on the coxa, there's a spine that you won't find on another species. Sounds a bit nitpicky, but it really helps you to identify just a few of these particular species. Now, I mean, this is probably the trickiest bit of with centipede ideas, just understanding these. But it's not that complicated. Possibly the more complicated thing, well, A, anterior, when you're looking at a leg that's facing backwards, well, which is anterior? You know, there is no anterior. But you imagine that the legs are, you bring the legs outwards, anterior will be at the front of that leg. So if you've got your centipede, you imagine that back leg sort of splayed out. It's the 15th leg, the anterior, the front edge of that, on the coxa, there's a ventral spine, which is unique, and that's your 15 vac. Yeah, I can't hear many of you thanking me for that, I shouldn't think, but nonetheless, you've heard it now. But there are spines which are helpful. It's the classic tale when you're trying to explain to people, you know, insects and invertebrates are not that easy to identify. Sometimes you might even be counting spines on the legs. It's centipedes, it's a classic. But anyway, hopefully that's straightforward. If you're lucky, it's Lamictes. It hasn't got any spines anyway. So hopefully that will be explained. And look at this slide again in detail, and that will explain in the future if you get stuck on how to understand that. I mean, they're interesting species if you're looking at those facts. So the soil centipedes, we're kind of running out of time, and I will go through this a lot. British geophilids have 35 on pairs of legs, as you know. Count the legs. First thing you do is count the legs. They're born with the right number of legs. All of the geophilomorphs have that number of legs from birth. So even if it's juvenile, it's almost freshly hatched, it'll narrow it down very much as to what you're looking at. As they grow, they're not adding any, but females may have a few more legs than males. So it might give you a clue sometimes as to whether you're dealing with a male or a female. On the very final pair of legs, again, you don't know how many legs, but it's not like it's the 15th pair. On that final pair of legs, there may be a claw, or there may not be. It may have like a little peg-like structure, like a stump with nothing at the end. It might just be a rounded structure, or it may be a claw. If there's a claw or not a claw, it's a very useful characteristic for you to look at. Another interesting characteristic on certain geophilomorphs is this, what is called a carpophagus structure. And around the sort of trunk area, if you could imagine it, sort of thoracic area, if it was an insect on its thorax, that end of the body. So around 9 to 12, 13, 14, the front end, but sort of between 9 and 12. there are structures on the sternites, on the sternum, a combination of like a little peg, possibly at the front there, a pit, or a group of pores. And the pattern of the pores, the size of this pit and the shape of this pit can be quite diagnostic. And there are pictures and keys to help you. I'm not going to go into details here as to what they look like, but these patch of pores that I've sort of drawn here, maybe a certain shape, this kidney-shaped pit might be quite distinctive. It might be a big semicircle. It might just be quite thin. But that's a useful character, and that's the carpophagus structure. And I'm sure we've got some pictures of that later. And then another really key characteristic for geophyllomorphs is on the last pair of legs, the coxa, that's this bit, the last bit that connects the leg to the body. There are various pores, and these little pore punctures, the position of them, the number of them, whether they're just underneath or whether they're around the top as well, very characteristic. So the coxal pores, very useful, but it's something which develops with age. So you might have the right number of legs to know it's one of these number of species, but it may have an immature number of pores on the final coxa there. So useful, but only in an adult. And again, they might vary slightly, but again, it's a useful character. Can be more readily seen if you've cleared the specimen. So if it's a dead specimen and you clear it with something like clove oil, just that's the easiest because it's harmless. It's stuff you put on toothache, buy it from a chemist. A little drop of clove oil tends to make your centipede go transparent, so you can see these pores. The whole room smells like Christmas, but nonetheless, you can see more of what's going on. But yeah, everything will smell of cloves for days, but it does help you to count the pore punctures. So those are the key the things: number of legs, has it got a carpophagus structure, what are these pore punctures doing, has it got a claw on the end of the last pair of legs. It's not many things to count, and then relate it to these charts of characters. Should be able to identify the majority of species. Some of them are quite small, so you do need to be possibly squashing between a pair of microscope slides, and that will help you see some of the pore punctures and the carpophagus structure a little bit more clearly. Finally, going to the scolopendromorphs, we only have cryptops species, and the majority, I think that there's a fourth one now, isn't there? But there are three that were commonly known. Hortensis is the one that's all over Britain. Anomalans is in the south, as is Parazygos, certainly across to Wales. But Parazyga anomalans tend to be more southerly. Anomalans is the big one that bit me. And you've got 21 pairs of legs and these big chunky back legs. And those are very useful. So the back pair of legs, they tend to curl up. If you collect one of these and you are preserving it, the sooner you unravel that back leg and let it sort of set and be preserved with the back leg out, it makes life easier, because otherwise they will curl in. And it's a gripping tool. As I mentioned, that large Scolopendra, the tropical one that caught the 3 insects, and as it passed it back, the last pair of legs were just like this, and they've got spines, little teeth inside, and they're really good for gripping. And some authors suggest that actually the back pair of legs in Scolopendromorphs are actually the thing they use for catching. But obviously they've got poison claws, so that's a very useful tool too, but they will catch things with the back legs. And I've been, there's a thing called Theotops, which is not a group of centipedes we get in Britain, but in Spain I encountered one of those, and I got attacked by its back end. It was sort of biting me, if you like, with its rear end, and this spine at the end, and these teeth were things that were grabbing. And it was, yeah, you didn't know which end to defend yourself from, really. So they've got these little teeth, and they're certainly very good at gripping things. I don't know what other functions they have, but the pattern of these combs, as they're called, can be very helpful for characteristics. So the number on the tarsus, the number on the tarsal comb, and the number of points on the tibial comb, and the structure of them are really helpful characters. You've only got 4 things to be comparing anyway with British Cryptops species, but that's a very helpful thing. It's slightly easier character is at the front end. On the head, there are little sutures, little lines engraved into the head, and they may be running just behind the antennal attachment. This picture is too pixelated to see, but there may be, there's probably one there and a little one at the back. So you've got a degree of incision of one sort or another around the head, whether it continues right across or whether it's just partial. In Cryptops anomalans, it's also handy in the first segment. So this part here, the first large, sorry, that one, this first tergite on the back has what's called a cruciform suture, and it's an X-shaped suture, often with a sort of dent in the middle. So that dent you can see. So it has a cross shape there. So if you find Cryptops anomalans, it's a big old beast anyway. So you probably know it's going to be Parasite or anomalans, and it'll have this dented cross just there, and there will be other little suture marks. But in Parasite and Hortensis, you don't have that cruciform suture. So when you read about cruciform sutures, that's what you're looking for. It's really a matter of getting the light just right, so it just catches that. So it's fairly straightforward. There are not many of them to mess with, and so there you are with your Scolopendromorphs. Now, just quickly, as I say, we're sort of effectively out of time, and I just want to show you some whole animals and some characters quite quickly. Just go through a gallery. I'll show you them each. So again, you can look at them at your leisure. Once this is on YouTube and recorded, you can pause this and have another look. But I'll go through them in turn without too much detail. But Stigmatogaster subterranea, I think it's now called Haplophilus subterranea. Again, it used to be Haplophilus, it became Stigmatogaster. I think it's now Haplophilus again. I may be corrected somewhere along the line. 73 to 83 pairs of legs. It's got a lot of pairs of legs. The key thing is on the hind coccyx, these paw structures, these little spots, are on the top and underneath, and they're sort of, not an infinite number, but you can't really count them. There's loads. So it's got lots and lots of little dots above and below on that final coxa. There are other, there's another Haplophilus Latina, which I've got pictures of I can show, which is much rarer and it's only in Cornwall. So I appreciate that people are joining me from all over the country and abroad, but most of these things are the non-littoral, the non-coastal species, and the more common inland ones. If you live in the south coast, you live on the coast, there will be different things which you may see. So you can see the colour. It's not got a carpophagus structure. It has no claw on the last leg. These are the sort of things you're looking for. So again, you're gathering clues and pulling it together. Another genus, Shendyla nemorensis. This is a fairly delicate little thing, pale coloured, quite small, no carpophagus structure. The cocci on the last legs have just got a couple of paws on each side. There's no claw on the last leg, but the last leg is quite swollen, so it's quite a big, bulgy last pair of legs. But again, the combination of the number of legs, somewhere between 37 and 43, and those other characters will narrow that down, hopefully, so that you can, and it's relatively distinctive, those last pair of legs, but it's not like they're the only ones that have swollen back legs, but this combination should help you. Henia, I think, is a much easier one, another genus, Henia vesuviana. So these are all geophyllomorphs, all very bendy. This is a big one, and it's this kind of olive, green, grey, it's a mucky coloured thing, really, often with this yellow stripe down its back. And it's quite chunky. If you find it, a really nice character that makes it quite distinctive is it'll curl into this ball with its underside facing outwards. And these little paws that they have on each segment are exuding a liquid, as you can see in this. So there's it's quite a sticky substance. So it's producing some kind of defensive structure there. And in order for that to be useful, it's curled up into a ball tightly with all its underside showing outwards. And if you find that, I mean, I found this one in Sheffield. It's not supposed to live anywhere near Sheffield, but once found, it's so obvious that this is Henia vesuviana. And yeah, very distinctive, but it can be a little bit more brownish, but basically it's somewhere between grey, green and orange. It's quite big. It's got 75 pairs of legs, no carpophagus structures, and quite swollen, fat pairs of back legs, with the paw, and in this case, it's very hard to see that there's a sort of pit that it's in, but you probably won't need to look for that because you'll have recognised it as Henia vesuviana. There's Henia, there's another Henia which I've got a picture of later, which is quite scarce, but you won't mistake it for this one. Another genus, Strigamia. The sort of common one, the woodland one, is Strigamia acuminata. It's a bright reddish coloured thing, as you can see, quite distinctive in that respect. 37 to 41 pairs of legs. Again, no carpophagus structure. You can look at the paws on the hind cocks are there, and there are 10 to 15. It has got a claw on the back of that last leg. And if you look underneath the forcipule here, the poison claw, there's another spine just there, and it's got a tooth at the base of this poison claw. So it's got this one spine there, and then it's got another tooth just there. So relatively widespread thing. You know, I found this in a woodland, and it's quite reddish. It's quite deep orange. So that should be fairly distinct if you find it. I'm not quite sure if, yeah. Another Strigamia, which, you know, we're very landlocked in Sheffield, so we don't see these. Strigamia maritima, maritime, it's a coastal thing. Turn over a big log, big rock on the beach where this thing occurs, and there may be loads of them. It could be like just masses of the things running around, and it's got similar characteristics to the last thing. It's quite a deeply coloured, quite dark browny orange, excuse me, you know, to quite red. quite large, 47 to 51 pairs of legs, only found in estuaries and seashores, and it's a bit more slender than the last one, which was quite fat and tapering at each end. Whereas this, this is, well, it's on the beach. There are other things that you could find, so do be cautious. I'm not an expert on coastal species, but this is one which is quite obvious when you see it, but do be careful. There are others that you might mistake it for. But again, this has got the extra little tooth on the poison claw as well, and the claw on the last leg. So Strogamia maritima. Geophilus carpophagus. I mentioned keeping voucher specimens. We used to think that there was a very variable thing called Geophilus carpophagus. It turned out that there was also a thing called Geophilus ethani, with actually a largely different number of legs. So there was the long form and the short form of Geophilus carpophagus, and only when research was done more carefully, we realized that actually there are two species, quite distinctive, different behaviors, and consistently shorter, well, fewer numbers of legs or more, and other characteristics. So it's a big old thing. This one climbs trees. So if it's up a yew tree underneath bark, which is a great place to find them in churchyards, it's probably carpophagus rather than ethani, which doesn't really particularly climb trees as readily, I don't believe. And it's just a matter of comparing the number of legs. So 51 to 57 for this, 47 to 51 in ethani. So you'll see there's an overlap at 51, which causes a degree of confusion. The last pair of legs for carpophagus, 4 to 8 paws, and there's 6 to 12 in ethani. But again, there's an overlap. So it's gather whatever information you possibly can. There are hidden paws. This one has been cleared, so you can just, so this has got clove oil on it, and you can see a couple of paws there, at least. I'm not sure how, it depends on your screen and how large you're seeing this. I'm seeing it quite large. But this is more associated with coastal areas, carpophagus, and buildings, whereas, and we used to record it around Sheffield, but actually around Sheffield, they turned out to be more ethani, up on the heathland, around Sheffield, up on the moors. The one that we thought was carpophagus originally turns out to be ethani. So 47 to 51 pairs of legs and 6 to 12 paws. So, and also you wouldn't tend to find this in gardens particularly, whereas you might find carpophagus in churchyards, gardens, and up trees. So it's quite clear. It's a matter of gathering your clues, looking at the comparison charts, and away you go. Final couple, I think, of Geophilus. Not going. We haven't got time. Flavus. This used to be called Longicornis, which was an easier name to remember than Flavus. So Flavus. It's quite yellow, which would generally, Flavus would usually indicate a yellow animal. When it was called Longicornis, it has long antennae. It's got long horns, Longicornis. So it's got quite long horns on it, long antennae, with quite elongate articles to each of them, which gives it its length. So it's quite nice. It's usually quite yellowish with a kind of red head, 49 to 57 pairs of legs, no carpophagus structure, and has a claw on the hind end, on the last leg. I say, I tend to think, oh yes, the picture perhaps bottom left there shows it's got really quite long antennae, but that's not utterly reliable, but you tend to immediately think that's what the beast is when you see it. So I preferred its old name. Well, yeah, I guess Flavus was its original name, and somebody called it Longicornis, but it's reverted back to its original Flavus. In Sculptus and others, it's a longish thing, up to 40 millimeters, and you're gathering information. This one, if anything, the character that perhaps I'd spot first would be that when you're looking at the pores punctures at the end, you've got a little bunch of perhaps 6, 4, 5, 6 here. And then where the red arrow is pointing, there's one sort of isolated pore on its own towards where it joins with the legs. It's not always easy to see, but if you clear the specimen with clove oil, you'll see a little bunch along this margin, and then an isolated pore at the other end. So it's got 4 to 7 pores, but they're arranged in an unusual way. It's got a claw on the last legs. It's generally pale yellow. The head's a little bit darker. But this one does have a carpophagus structure. So when you look underneath at sort of perhaps around 9, 10, 11, you see this big sort of crescent, well, semicircular pit. It'll have a little peg here, and there's a pattern of spots and pores in the center, which are also part of this pattern. You can't see it very easy. You can't always get the light in the right in the right plane to see all of those structures at once. But yeah, the shape of that is very helpful. I'm not sure if we can show you a different one. Yeah, no, not really. Anyway, so Truncorum is very small. Underneath, if you looked at its carpophagus structure, looking at the electron micrograph picture, the thing I showed you as a live video earlier was this. It's small, it's quite delicate, fairly pale, but where the carpophagus structure is, if you just kept the light right, and I did it with a hand lens, I could see it. It's got these 3 depressions, so longitudinal depressions on each of these trunk segments. So there's a dent there and a dent there and a dent there. So if you get it just at the right angle with the light from the side, even with a hand lens, you can see these little dips. So that's helpful. So it's also got various carpophagus structural patterns, but others don't have those 3 side-by-side indentations. 2 pores on the coxa at the end, and it's got 37 to 41 pairs of legs. So, but it's quite small. It's usually just over 1 centimetre to 1.5 centimetres. It's not usually 2 centimetres long. So, and very soil based. It's very much in the soil. This thing underneath logs, just into the soil. Right, that was your geophilids. We're nearly there. 3 cryptops. I've mentioned them already, so I won't go into detail, except you can just see them a bit more easily. They've all got 21 pairs of legs. Anomalans is big. It's up to 5 centimetres long. It's got this cruciform suture on that first segment of the body there after the head. So that's nice and distinctive. From this angle, you can't see it on the bottom picture, but it's a big beast. That's my thumb. It made a nice pet for a while before I released it back to where it came from. And big chunky back legs, which will have distinctive teeth of one sort or another. The comb has 7 to 10 teeth on the tibia and 3 to 5 teeth on the tarsus of the hind tibia. So again, you'll that's what you're looking for. But again, don't worry if you need this information, just wait till it's on YouTube and you'll be able to pause this screen. But so that's Anomalans, big one. Hortensis, the little one. This is less than 3 centimetres long. All got 21 pairs of legs, of course. It's just got a short suture on the head, just a little one. The comb on the back leg, 5 to 8 on the tibia, 2 to 4 on the tarsus. You can see on this central picture how if you don't unravel the leg soon enough and it's pickled and dries up a bit, you can't see these combs underneath because the leg is folded round on itself horribly and very hard to see these structures inside. But you can also see how it's very good for gripping things. So that's the smaller one, and then the middle-sized one is parisi. And you see, I managed to photograph this one as it ran over the corner of something. So its leg was sticking out beautifully, and you can see all the spines and the combs on the underside of that hind leg, 6 to 12 on the tibia and 4 to 9 on the tarsus. And the suture is at the front and the suture is at the back of the head. So there's a little bit there and there's a little bit there. But they're sort of, yeah, intermediate. Some of you live in areas where probably all three of these occur. Around here, we only really get hortensis, and then that one rare occasion where we got anomalans. So it's easy for me in Sheffield to think it's just hortensis, but some of you it's just a little bit more, a bit trickier. See the picture at the bottom there, was one has escaped. That as it tried to get away, its back leg came off. So you can appreciate how that back leg would be really useful to me if I'd not managed to catch the thing, because I would still be able to look at that back leg and identify this. Finally, the Lithobia species, the Lithobia morphs. Yeah, I won't go through every characteristic, but we're looking at how many antennal segments, so and how many ocelli on each eye. Are there any projections along in the back corner of sort of 7, 9, 11, 13? How many of these little teeth are there underneath its face on this coxosternite? And are there any special little spines? This one has a special little spine. Borealis has between the 15 VPP and the 15 DPP, it's got another one. So on the 15th leg, between the ventral and dorsal posterior prefemur, there's an extra spine, which is nice to know. But hopefully you might have found that counting the ocelli, counting the antennal segments and finding the projection on 11 and 13, tergite 11 and 13, should probably have told you sufficiently what it was. Occasionally this one's got a projection at the rear of 9 as well. But if all else fails, it has a unique spine, which is quite helpful. And it's there. I think it's in that picture, but it's no, where are we? What are we on? Prefemur, so yeah, it's there. Anyway, we went labor. Lithobius calcaratus. You might think that calcaratus refers to the fact that it lives in calcareous soils. It doesn't mean that at all. It means that it has a calcar. So on its last pair of legs, it's nice when they've got something unique. And this calcaratus has a unique character, which is great. You can't quite see it on here, but this male, it's a male. I didn't go into telling the difference between a male and a female, and I don't think I will now. But males and females have different structures on the very last terminal segment of the body. This is a male, and you can just see a bump that, again, depends on your screen and what the resolution's like. But just there, and just there, there's a bump, and it's arrowed here. That there is a very distinct peg. So it bows out like that, and then there's a peg. And that is a calcar. That is known as a calcar. So it is Lithobius calcaratus because it's got a calcar, this little extra peg on a male 15th leg. That's distinctive. Otherwise, you're still counting antennal segments. You're still looking at how many ocelli. It's got up to 9. So it's not got a lot of eyes, this thing, and just 2 forcipular teeth on each side. But it's nice that it's got a distinctive additional thing. The most common one you're going to find is Lithobius forficatus. It's big, it's chestnut coloured. You'll find it in all sorts of stages of age. So counting its antennal segments and its ocelli and its eyes, may not be that helpful ultimately unless it's an adult. But when you get a big one, getting on for, you know, between 2 and 3 centimeters long, you'll probably be getting on for adult. It probably have, you know, up to 40 eyes. It'll have, up to 40 antennal segments, and it'll have loads of eyes. The ocelli will be large in number, but up to 30. So you look, and the key thing really is that if you flip it over and look at its forcipular teeth, it'll have 5 to 7. So it could have 5 plus 5, it could have 7 plus 7. So it'll have a good number of those. But on the back, if you count along the tergites and look at the corners, it'll have projections on 9, 11, and 13. So it'll have projections there, there, and there. And it just have a single claw on the last leg. Each of those is quite crucial in that there are other species which are quite large and red, which you might just, I think, names escape me, Peregrinus, Pisceus, various things which could also look like that. So again, however easy you might think it is, you'll be wanting to gather the information on antennal segments, points on the corners of the tergites, number of eyes, forcipular teeth, and come up with an answer. Massilentus is a nice little medium-sized thing. Find it under limestone rocks all over in the Peak District and around Sheffield. It's I've seen it. 39 to 45 antennal segments, antennal articles, up to 9 ocelli, so not many eyes. But this one has it's got projections on tergites 9, 11, and 13. So you'll find it's got pointy bits. It's got 2 claws on the 15th leg, and it's parthenogenetic, which means they're all female. I really would love to go into the details that a lot of centipedes, if you look at a map of the centipedes around Sheffield when I was mapping them, I could tell which ones were the parthenogenetic species to an extent, in that you'd get a little group of records and another little group of records and another little group of records, as if one had arrived and it only needed one to breed. So it had sort of bred and dispersed itself outwards from a single point, and another had done the same. So these parthenogenetic species on maps can often look in little clumps, as if one individual's arrived, because it's not had to wait for another male. It's not had to arrive with a friend before it can reproduce and have males and females. One can reproduce, the next one can reproduce, the next one. So you get these nice little clumps of records, and that's Massilentus around the Sheffield area shows that quite nicely. But yeah, this one's got 2 teeth, 2 claws on the 15th leg. So you just, again, combining characters to there are 1 or 2 other special things when you start looking at these things. Melanops is the other thing on the video earlier. It's got a dark stripe down the back. It's not the only one, but its back legs are not stripy, but it's got a stripe down the back. It might not run away quite as readily as some of these earth centipedes. And it's got projections on tergites 9, 11, and 13. It's got 32 to 42 antennal articles and 2 or 3 forcipular teeth. And again, 2 claws on the hind, the last legs. But it's quite light coloured, and with this dark stripe, but no stripes on the legs, makes it look reasonably distinctive with experience. I'm not sure if Variegatus is next. It's not, but when we get to Variegatus is bigger, similar, dark stripe down the back, and also stripy legs. So, right, this is dragging on. Sorry, folks, I'll come to a conclusion. Muticus, same character, group of characters. You're looking at various things. When you're looking at the forcipular teeth, This is one that's got quite sloping shoulders away. It's got shoulders as opposed to just disappearing off at the edge. So you've got the 2 teeth and then quite a shoulder, and quite blunt projections in the corners of the tergites 9, 11, and 13, and just a single claw on the last leg. If you've got a male, the face is quite sort of chamfered, sort of I don't know, it's a bit more pointy at the front, and so it's a little bit more forward facing. And also, if you've got a male, again on the 14th leg, there's a swelling, which you can see it when you've got one. But if it's quite forward facing and it's got this additional little swelling on the 14th, on the tibia of the 14th leg, you'll recognise that. So again, odd little unique things are quite helpful. Pilicornis, I thought I'd found a really big Forficatus once, and it was very big. It was Pilicornis. This thing will bite you. Won't do you any harm, probably won't pierce the skin, but it could potentially. It's got a big old pair of jaws on it. Up to 35 millimeters, quite red. It's big in every department. It's got a lot of antennal segments. It's got a lot of ocelli, and but it's only got 3 to 5 falcipular teeth. So it might have a reasonably small number of those, but it could have 5 plus 5, so it could look like Forficatus. But it's only got projections on tergites 11 and 13. So when you come to look at it and you think, oh, it's Forficatus, by the time you've counted its antennal articles and its eyes and found these projections that are not there on 9, and look, it's got 1 claw on the 15th leg, and you'll realize it's not Forficatus and know why you got bitten, because it was Pilicornis and it's supposed to be that big. Which is not to say you won't find big Forficatus, but not that big. So it's a lovely thing. I think it's in London, down south on the coast, or quite far, quite near the south of the country. But it was in a cellar in Sheffield. That's where I saw it. And the classic Variegatus. If the one thing you learned today, it's this: recognise Lithobius variegatus. It's probably got a stripe down the back, or at least sort of a very acute triangle of dark on each segment. It's got black and white or yellow and brown stripes on the last few legs in particular. Usually has very pale and broad falcipules as well, which is not unique as such, but it usually looks like it's got very big jaws and fangs. It's got tergite projections set at 7 as well as 9, 11, and 13. It thinks it's disguised because it's sort of slightly camouflaged, so it doesn't run away. Tends to sit still. It will run away eventually, but tends to sit still, and it's pretty big. More in the west than in the far east of Britain, but it's pretty much everywhere, and under most logs in woods around here. So do record that one. Lithobius variegatus. It's variegated. It's got stripy back legs, probably a stripe down the middle, and it might not run away, and it's big. So record Lithobius variegatus if nothing else. Crassipes, it's got 20 antennal segments. This is what I've already described. In Curtipes, which is the next one, Curtipes and Crassipes have 19 or 20 in Curtipes, but always 20 antennal articles in Crassipes. The Crassipes curls into a ball when disturbed. Sorry, it does not curl into a ball when disturbed. The Crassipes remains like it is there, it's running around. If you find Curtipes, it's curled up. So if you find a centipede that's curled up, make a note in whatever form you wish. So when you come to look at it, you'll remember that it's curled up into a little ball, just curled up on itself if necessary, because it's quite helpful to recognise a couple of species. Curtipes is also the one where there's one ocellus and then a little rosette of eyes around a central ocellus as well. No projections on the tergites with these, not many eyes, but they are arranged in a particular way. So again, I'll go back up so that's been seen for a moment or two for when you come to look at it again. The thing with Centipedes is also on the 15th leg of a male, there's this bulge here. So on the it's the tibia, is that the tibia? Yes, it's tibia. There's that bulge at the rear end, which is distinctive and unique. So you got the right same antenna. So 1920 antennal articles and that bulge, and it curled up when you found it. That's Centipedes. Microps will also curl up into a ball, but it's little, as the name suggests. But it's easier to recognise in that it's got just 3 ocelli on each side. So just you look at the side of the thing, it's probably got, it might have 4, but generally 3 on each side. 3 on each side, curled up into a ball. It's quite little. Usually 2 claws on the last segment, but you know it's never even a centimetre. It's a little thing. So that's relatively straightforward to recognise microps. But again, try comparing all of the characters until you're familiar with the beast. I'm never quite sure whether I dare say you can record that in the field just from glancing at it. But I think if you can see the 3 eyes and it curled up into a ball, Tony may tell me I'm wrong, but I think I'd call that microps if it was me. And then finally, Lamictes imaginatus. It's only got 1 ocellus on each side. It's got no spines on its legs. It's got 3 claws at the end of the 15th leg. They're all female. This one shows a beautiful pattern of distribution in the Sheffield area where it's found. 3 plus 3 falcipular teeth. But really, it's quite dark. It's got 1 ocellus and no spines and 3 claws at the end. So Lamictes imaginatus is a pretty distinctive creature too. And there's our old friend Scutigera, which you'll only rarely see in Britain, but compound eyes, fewer tergites, enormously long legs, which will come off in your hand, violet grey. It runs like crazy. The last leg has over 500 segments. There's so many little articles to the and each of them have got little hairs underneath, so it's very good at gripping surfaces. So its legs are not like most centipedes are running on their tiptoes and with their claws. This is running as some insects would, with several articles on the ground, each with little spines. So it's very good at gripping and very good at terrifying you as it runs across your living room when you're on holiday in continental Europe or Jersey. So there we go. So I'm going to, that really did go on longer than I'd hoped. So apologies for that. I can close this thing down. And return you to this. So the last 2 of you that are still standing, or however many are still out there, there are some rarities which I may all Henia brevis, Plephilus sulatinus, with lots of spots on it. So there are a range of other things with very distinctive spots underneath. This is a thing from a hothouse, Chicarup javanicus, with a very long sort of very red head, very long forcipules there, but only in heated greenhouses. What I want to just finish with this, whoops, I want to finish with just that, which was the main points are all centipedes have an odd number of leg pairs. The Geophilum also got 35 to 100 pairs. They have all their legs from birth. Scolopendra have got 21 pairs and all their legs from birth. The Lithobiomorphs have got 15 pairs, but the young have fewer. Scutigeromorphs, likewise 15 pairs, and the young have fewer. And again, you can look at this later on screen, but I've summarized here that the geophilomorphs, the coxal pore number are increasing, but all their antennal segments remain the same, and they look after their young. Scolopendras, coxal pore number increases as they mature, and they've got various number of antennal segments, and they care for their young. The stone centipedes, the thobius, their ocelli number increase, the antennal number increase as they grow, and they just throw out their eggs. And I don't even need to, it's grayed out somewhat at the bottom because honestly, you're not going to mistake a scutigeromorph. But that's on screen for you to summarize some of the basic facts.
So from Joanne Church, you said that centipedes have poison claws. Would they make you ill if they bit you?
They would, tropical ones that are big enough to pierce human skin and have a reasonable amount of venom, they do make people quite, you know, it's like a hornet sting in that you know, it's like a bad wasp sting, bite, if you like. In this country, I don't think there's even an issue of anybody ever getting an anaphylactic reaction, as far as I'm aware. I'm open to correction on that, but even when you scour the literature, even internationally, there just aren't even really deaths. There are a few, and I say, but there's in sickly children and things like that, and I suspect there are probably small dogs or whatever. But it's not pleasant. There's localized swelling and a lot of pain. I mean, I've not been bitten by one apart from the that anomalans, but it did make a little swelling. So that there's a reaction, and I could feel a numbness. But generally, in Britain, they're just not big enough to puncture the skin. Apart from the Thobius pilicornis, probably maybe there's probably a couple of other big ones out there that might bite you, but it's not. No, they're barely like a wasp sting here. But the big scolopendras internationally, I would avoid, really.
Yes, yeah, especially from your account with the crickets.
Yeah, absolutely. Yeah, as they grab each of your children and pass them down. Yeah, and eat your children slowly throughout the day afterwards. Yeah.
Okay, so a question from Kate. Is the CD available as a download, or could it be emailed? I don't have a CD drive.
It's not at the moment, no, and it won't be just yet. We've still got hard copy CDs that we'd like to disperse first, but what has been recorded here is as close to that as you'll get, because this is on YouTube. Each of those, you know, I've been at pains to sort of show every slide. Possibly some of them were a bit swift today, but so that on YouTube you can pause them, and they are effectively the same as the CD. But it's nicer to have the CD. It's got some elements of interactivity in there, not much, but it will send you back and forth, certainly in the Myriapod key. So it is worth having the PDFs of that, because there is an element of interactivity, and you get the Myriapods and you get the woodlice. But for now, because there's no in-person activities, yes, you've got to buy the CD from Paul Harding. All the details are on the website now, on BMIG's website. So yeah, it's part of the reason we bought them from Nature Bureau, is because, you know, few people really have a CD drive anymore. So we want to distribute them more quickly than and basically get them out there while people can still use them. But ultimately, well, there will be a version online, but not in the next year or two. But use this YouTube channel.
Okay, great. A question from Kia Flowerdew. when I've collected centipedes in the past, they have become quite brittle in the alcohol and often set into quite contorted positions. Is there anything I can do to combat this?
Some people put glycerol, some sort of, if you can get glycerol and put it into the alcohol, that means that it's not so much that they remain more supple, but it means if they dry out, it's easier to re-preserve them, so there's still got some flexibility. It makes them a little bit more flexible. It saves the joints becoming really brittle, as far as I understand. The alcohol, the higher the alcohol concentration, the more brittle they get. So 70% will produce a more flexible specimen than a 90% alcohol specimen, and pure ethanol one will snap like a twig. But
It'll be good for DNA.
Yeah, but great for DNA. So the other thing is, if it's quite a long thing, I mean, some people put them in straws. If you really want, what you ideally want is a long elongated one. So if you've freshly killed one, and I know often, you know, myself included, you collect it and then you look at it days later and it's a ball of a knotted ball, which is a bit tricky, but they're still usually flexible. But if you're looking at it almost on the same day, and you get it on a microscope slide and straighten it out and have a look at it there and then, they will stay a bit more elongate when you drop them back into the alcohol. Or if it's already on you perhaps got it on a microscope slide straightened out, then either cut a strip of plastic plastic straw, drinking straw, and sort of lie it against it and pop that back into the tube. Or better still, if it's a small drinking straw. The reason I'm talking about stripping it is because they're too big for a centipede, but if you cut a slit in it, you can then make the drinking straw narrower and retain the thing in a position which is more elongate. But this is largely unnecessary if you just stretch them out while they're relatively fresh, and tweak the legs out, and then preserve them without just throwing them in and leaving them. Hope that helps.
No, really useful tips. Okay, a question from Graham Smith. Where is it best to get increasingly finer grained sieves for sieving through leaf litter? Would be nice to get some standardized ones.
The ones I showed in the picture cost about £100 per sieve. They're ridiculous. They're sort of the high grade analytical sieves. They cost a fortune. So don't go there unless you work in a university. What you want to do is search for, what's the word I'm looking for? What are those little trees? Bonsai trees? Look for websites for bonsai enthusiasts and buy a set of sieves from them. They must sieve the soil to precise levels and scale their soil to their trees or something. I've got a set of bonsai sieves, and they work well because there are all sorts of different grades. I've also got a set which have got interchangeable drop-in gauges of sieve. If you just look online, so I've got like garden sieves which have got 4 different gauges from like a centimetre down to about 2 millimetres, and that's the easiest thing. You can, and if you want to sieve from one to the other, then you have to buy 2 sets of those that sit on top of each other. But I change the gauge of my sieves according to what I'm looking for. If I'm after springtails, I'll use a finer one. If I'm after centipedes, I'll use a bigger one. But there's a lot online, and the interchangeable ones. But if you need a clue as to what brands, I think the bonsai people are the people to search.
Well, I've never heard of bonsai sieves. So excellent. Okay, so a question from Veronica Pazikaina, apologies for the pronunciation there. Are there any features that can tell us that a centipede is a young or immature individual?
In my opinion, I would say I'm not entirely sure. There are genitalia structures on some of the females, and which won't be there. But I haven't gone into the details of distinguishing males from females and talking about the various gonopod spurs at the hind end. So it might be difficult for me to answer that without going into quite complicated things. But some of the signs of maturity in the genitalia won't be there. I just tend to be counting things and suggesting and just questioning whether it's adult because it hasn't got the right number of features. I can't think of quick, easy answers to that. I mean, obviously, if they're immature or they're not fully hardened and they're still purple and soft and you know, recently molted, that doesn't necessarily mean it's not mature, but it shows it's still in a period of its life where it's still shedding, possibly regularly. So I'm not quite sure. I mean, unless I don't want to draw in Mr. Barber at this point, but if anybody out there knows better than I do as to how to recognise whether something's immature, I'm happy for anybody else to make a suggestion other than counting things and thinking it's probably not adult yet.
There is a Tony. I don't know if it's.
Yeah, I don't know. I don't want to point out your presence in any way. But I can't. I mean, unless anybody else has got. To me, it's not like they look that different. It's just that there are features which are not yet there in insufficient quantity, and as I say, and genitalia structures won't be there. But I can't. I don't really. It's not something I've had to deal with as such, and you just despair. You just think, this one's not keying out. It's probably too young. That's not the most helpful answer, I know, but it does make it a bit tricky when they're immature.
You can do geophyllomorphs very easily. Can you switch me on?
I can hear you, Tony.
Yeah, right, you can do I was trying to keep right out of this.
Yeah, I'm sorry to mention you were there.
Congratulations on doing it anyway. It was good to hear it done well.
I learned from the best.
Geophyllomorphs, you've got the number of legs is the same, so it makes it very easy. You don't have to worry whether you've got a juvenile or not, actually. Lithobius, all the later stages have the same number of legs and most of the same characters. They're just not all well developed. It's the thing you pick up with experience, actually. And there is the thing, if all else fails, send the specimen to Paul Richards.
I don't remember that one. That's a new technique on me. Yeah. I think I'm sure I used to send mine to you, but there we go.
Thank you, Tony.
I'd say I didn't think there was anything. There's nothing blindingly obvious, but you know whether you've got the right number of legs and stuff.
What you don't have to do is what you have to do with millipedes is look for the genitalia, because they're really not very helpful anyway.
No.
It's nice to work with a group that doesn't have that problem. So
that is good to know.
Sorry? Yeah,
that is really good to know. No genitalia.
I mean, you can do nearly adult ones, often juvenile ones, and you can do females as well as males. In some cases, females are actually easier, but it means there's not nice, simple picture of sort of the naughty bits like you do for a millipede, which you can compare.
Yeah, that's part of the reason I didn't go into the male-female thing desperately much, because
it's complicated.
And they all look pretty much. There are spurs and a number of spurs and slight changes and variations, but as Tony says, it's not like with a millipede where you get a decent male with a really nice gonopod, and you can identify your millipede pretty much just on that. It's not as helpful as counting its antennal segments and looking at the paws and looking for projections. So it is quite nice in that respect.
Actually, one piece of advice I'd always give is, if you can't identify a thing, keep it if you can, and preserve it, and go back to it one day.
Yeah.
If you haven't got Paul Richards to do it for you.
And he won't be here forever, you know. Well, actually, I've captured now, aren't I? I mean, it's on YouTube now forever.
Yes.
There's no getting away from me. Woodlice next, folks.
Okay, I'll go on to the next question. This is from Angela L. Should we use the name G. insculptus or G. alpinus?
I've never had an attempt to answer that one. Where are we at? What's it currently known as then, Tony?
Alpinus.
Right.
I was going to do something and say that actually you should use something like, I forget what it is now, CEN 123 or something, which is the sort of species code. It is Alpinus now. But I mean, actually, I don't think you should worry if you're recording it, because both names would still be recognized. And I think that's true of any of the others.
Yeah.
Names do change.
And the names, it's a problem. We could get hung up on all sorts. I mentioned Stigmatogaster becoming, or Haplophilus becoming Stigmatogaster, going back to Haplophilus. And, you know, names do change, and, you know, you can only do with what's presented to you, and what your publication in front of you says. You know, I'm still calling Microps dubosqui, I think. I don't know. You grow up with certain names, and there are people that never know that names have changed anyway. So hopefully these days the systems out there are such that they recognize synonyms reasonably well. If there's a specimen, it doesn't matter, because we can go back and look at the specimen.
Yes, I think that's it. And there are very few centipedes, British ones, where there's any likelihood of confusion. In other words, the same name hasn't been used for two different species. There are one or two, but generally either name would be accepted.
I mean, when I found that one I videoed earlier with the sort of three gutters across its segments, I thought, oh, I found Brachygeophilus. I'm still calling it Brachygeophilus, because to me, brachy means a short thing, and then this was a short thing, and I knew it was a little. To me, it's like Flavus isn't as helpful to me as Longicornis. I found the old names told me something about them, but you know, I'm learning. Even at my age, there's still room in there to teach an old dog new tricks.
Stigmatogaster has changed its name to and fro about 3 times in the last 20 years. It's now Stigmatogaster again.
Is it? Great. Great.
In a forthcoming French publication.
Okay.
Okay, we'll go on to the next question then, from Steve Orridge. Do centipedes have parasitoids?
My answer to that would be almost certainly, but I don't know enough. I don't know anything about that. But my knowledge of parasitoids is that they just do everything. I mean, I can't believe there isn't, but maybe there isn't. I don't know. I'm ignorant on parasitoids, but I do know that whatever Hymenoptera, Diptera I've studied about parasitoids, it just seems to be something for everything. So I know millipedes have got them, you know, woodlice have got them. So sure, I don't actually know. I'm happy to say I assume so.
Yeah, there are some protozoan parasites and some worms. There's quite a lot of things. I mean, I don't know much about them, but and I don't know anyone that's done any recent work on them.
That would have been a good question for Gavin Broad last week on the nocturnal ichneumoniid.
Yeah.
I don't know if there's any of those on centipedes.
I suspect that because they're a fast predator, I suspect that the sort of dipteran,
Hymenopteran
type of parasitoid, they're probably not going to have a life history whereby they shoot in and lay an egg. I could imagine their larvae might be in soil and grab hold of the leg of a centipede in the same way like more microorganisms would. I can't imagine it being an easy thing for a wasp to locate a centipede and insert an ovipositor, but I could imagine it laying an egg that's free living, that is picked up in it, or it lays them into their eggs or something like that. That's a completely different subject. It fascinates me. I find parasitoids are fascinating. They'll have a strategy for it, but I suspect it's not chasing centipedes with an ovipositor.
Yeah, I suppose they don't have a soft-bodied stage, do they? Really?
No, you've got to get it between tergites or down the side of the pleurites, and who knows? I mean, you slap one in its trachea and it may well be away, but it's gonna fight back. It's an animal that's gonna fight back. Whereas inadvertently picking up some kind of larval parasite which wheedles its way in is probably much more the mode of parasitism, I would have thought. But don't know. It's fair to say, I mean, Ethan wrote that amazing book with so much information, but I say you read through it and there's so little known about a lot of these things. I'm very impressed with folks who work on centipedes in that there's not a lot of information. There's a lot that's not known about their reproduction. This is quite difficult stuff. And so moving beyond their reproduction and their life histories into parasitism as well is a step too far for me, but it's some way down the line in a group of organisms which have still so much to learn about. So the more distribution knowledge, the more people can get to recognize them and get enthused by them and maybe get into looking at them, the greater the knowledge bank will be. So get out there and embrace the Chilopoda. Somebody out there, we need more people looking at them.
Absolutely. Okay, a question from Tammy Horton. When is the best time of year to look for centipedes?
They're around all the time, but I personally tend to look in. They don't mind it so dry as millipedes, so the summer's not as bad. The spring autumn's easiest. They're less active in the winter, but you still find them. But spring and autumn, I think because they vary in their life histories, I'm not sure whether I would lean one way to spring and one way to autumn. But spring's very good. I mean, go looking in March, April, May. I would have thought would be a good time, but they're around all the time. But they're reproducing spring, summer, and autumn. And they're not doing much in the winter. So, but because of the damp, they like damp conditions. The things they're feeding on are more prolific in the autumn and the spring. That's always with these things, always with the predator. I think, well, when's its food supply around? So I would say, when are the woodlice around? When are the springtails around? Well, they're around all the time, so they can feed on them all the time. But spring and autumn are just damper and more temperate. So, yeah.
Okay, well, good timing for this webinar then. People can start to think about going out and looking for them. So possibly a final question here from Ben Hargreaves. Any habitat specialists? I know a few were mentioned, e.g. peat bogs and limestone pavement.
In all honesty, I'm not enough of an expert to pick up on many of the, and a very good answer to that. But there are lots of coastal ones, and there are a bunch which are a lot of quite scarce ones, which are, you know, they're in Cornwall, and the ours are silly and things. They're potentially, there could be all sorts of climatic reasons for that, but and also there's a whole bunch of coastal habitat reasons for that. The reason I'm a bit vague on a question like that is because I only deal with landlocked Sheffield more than anything. I've recorded them on the coast. There are things which climb trees. We mentioned like Joffa's carpophagus. You know, if you want to find that, look up a yew tree is a very good thing to look for carpophagus, because it's very easy to climb, but they'll climb up there. And so there are some which will climb. There are, I say, Aethani around here, the equivalent of carpophagus, is much more an upland, doesn't mind the acid, peatlands, and so there are, but to be honest, I'm a bit ignorant of what some of the southern ones do and what the coastal ones are, but there definitely are other ones that, other than Shindala, the marine, the Maritima, the coastal one. There are other coastal species. So, you know, splitting rocks on a cliff safely with a hard hat on, obviously, health and safety and all that. But yeah, breaking rocks on coastal cliffs is really good for finding. That's where I found Henia vesuviana quite often. There are a number of things which, yeah, are more coastal, but that's as close to an answer as I think I can accurately give. I'll start making it up after that.
Thanks. Well, there's plenty of coastline to work in.
Yeah.
In our Cheshire, Lancashire area, and I think Ben was thinking of really the Morecambe Bay limestones area in North Lancashire.
Very difficult. Areas. Very difficult to sample limestone pavement.
Yeah.
Yeah.
And wet moor doesn't yield very much, but it does occasionally. Salt marsh with rocks on it, with bits of stone on it, might turn things up. But the best place to look is in your back garden.
Yeah.
In Nepal. And basically, it's urban areas. And
in my back garden.
People's back gardens. Maybe not yours, but it is.
No, my back garden's rubbish. Yeah.
Yeah. Yes. And if you want to attract them, just dump rubbish there.
Yeah. Yeah. I'm not an advocate for tipping, fly tipping, but fly tipping, a lay-by fly tipping, because you get everything from everybody's gardens. If you want a variety of centipedes, go and look at some fly tipping. It's just true. There's a lot of quite synanthropic, you know, the Henia that I found in Sheffield was definitely imported by somebody into the bottom of their garden. You know, the anomalans, the Cryptops anomalans, the big beast, that's not round here at all. It's miles from it. It's, you know, native. There are a whole host of and in cellars, all sorts of things. So very much urban. Urban's good. Urban's good.
Cemeteries. Cemeteries and churchyards.
Cemeteries, absolutely. And that's, you've got your yew tree, so you can find your carpophagus up it. I found the Peak District limestone dales, I find different things there. To, you know, I quite on fairly well embedded rocks in grassland in limestone dales. I find they're very good for little dark, annoying little centipedes. Lots of little dark centipedes about that big, which you have to have a close look at because they look a bit similar. But you know, I found all my Calcaratus muticus, kind of the medium-sized things there.
If you want a challenge, there's 3 or 4 species of about 1,000 meters up in Snowdonia.
There we are then.
On the rock scree.
That are only in Snowdonia.
No, they're not only there, but you know, we've just discovered that there are about 3 species that seem to be at that height, which is a pretty unusual height anyway for Britain. And why they're there, we don't. Well, obviously they can tolerate it. And
Yeah.
it's not a site that most people go to because you've got to climb a mountain to do it.
Yeah.
I've certainly seen variegatus on the tops of the fells in the Lake District.
Yeah.
Turning over rocks there, yeah.
Variegatus Curtipedes in Wales, on the mountains, and one of the Stragamias turned up at 1,000 meters, which is quite a height for centipedes in Britain anyway.
The best answer we can give, of course, is you should be recording everything everywhere. So we just want to know, just wherever you, whatever habitats befall you, record the centipedes there and the millipedes while you're at it.
Okay, well, I think we'll leave it there then for this afternoon. And just thank you so much again, Paul. And I'm sure there's so much information that everyone is probably going to have to go back and go through the YouTube video, which is just brilliant that we were able to record it these days and have it for people to use. So that's fantastic. And we'll see you, Paul, very soon, and hopefully everybody else who's still here. And yeah, and thanks to Leanna for doing all the organizing and marketing and getting this, making this happen. So yeah.
Thank you very much, everyone.